So, our next speaker is Professor Praveen Karanth, and all that I said before about 42 and all is like not true. We're basically commemorating 20 years of Praveen being in CES.
Yeah, I think Janvi basically has stolen my thunder. All the cool stuff she's already talked about, contributions by the researchers in CES to the larger data set she's already talked about, the overall framework from microevolution to macroevolution she's already talked about. So I don't know what I'm going to talk about. So, I did check, but still. No, what I should have done is I should have gone first. Okay, so, was that? So, I'll just summarize the evolutionary research going on that we have undertaken here in CES. Obviously, there's a lot of work that has been done. So I'm just going to pick a couple of interesting studies by various faculty and sort of talk about those studies. So the title, am I audible to everyone now? Is it okay? Okay, so the title of my talk is Micro to Macro Evolution in Space and Time. But before I sort of jump into it, I'll just quickly go over so that we are all on the same page. So basically, I'm going to talk about microevolutionary studies that we are undertaking here in CES, also microevolution and biogeography. Microevolution is basically, within species evolutionary changes in allele frequencies or change in trait value within a species across generation. So basically within species, how genetic diversity is partitioned, how it changes over time, and so on. And you have areas like population genetics, molecular evolution, phylogeography, that sort of largely falls under this area. Macroevolution, as you know, is speciation in everything above, right? So processes that generate species, we are looking at things like drivers of diversification. Are they key innovations that drive diversification? Adaptive radiation is a classic case of macroevolution. And a lot of our work has been in the area of biogeography. In biogeography, we are interested in looking at processes that generate spatiotemporal patterns of biodiversity distributions, right? And particularly how past events have shaped current distributions. So that is where we are at right now. So a lot of the work is in the area of biogeography, but in fact, biogeography, some people would consider it part of macroevolution, right? But I've just shown it as separate. All right, let's start with microevolution. This is work from Guttal lab. So there'll be a lot of terrible equations, no pretty pictures. No. I've tried to make it simple. In fact, he made this slide for me, and I then had to scrub it to make it palatable. All right, so typically what happens when we go from one generation to another generation, you have a population of individuals with different traits, and there's birth and death. And across generation, the frequency of these traits change, right? And what brings about this change in frequency? Well, it's selection and drift, right? And selection happens when one of those traits has higher fitness value than the other trait. And over time, that particular trait then gets fixed in the population. So in this case, the blue trait has higher fitness value. So it gets fixed in the population. All right. So that's in a very, very large population. But if you have a finite population, particularly if you have a small population, And if the fitness values of the two traits are the same, you have drift, right? Either one of those traits can get fixed. Uh, what we are, you can look at it as traits, you can look at it as alleles, and either of the two alleles can get fixed, right? So this is in case of a small population. And the fitness values are the same for the two traits. However, one of the fundamental assumptions that these models make is that population size is fixed, right? So you're not perturbing the population size, right? But in nature, stochasticity results in fluctuation in population size, and that means the frequency of those traits or the alleles are also fluctuating, right? So the classical models ignore that population size fluctuates across generation. It just assumes that it's constant. Now, this is a very, very important point because once you incorporate this stochasticity in population size, you begin to see something very interesting. So again, going back to our classic situation, one of the trait has higher fitness, and basically you expect it to get fixed. The assumption here is the population size is really large, ideally infinite. However, if you have a small population where population size is changing with time, right, then blue may be preferred over red. So in this example, the red allele or the red trait has higher fitness, right? So you expect the red trait to get fixed in the population. But what Goethel's lab's work has basically shown that if the population size is small and if the population is fluctuating, then you have this very interesting scenario which they called noise-induced reversal of selection, right? So instead of the red, the advantageous trait or the allele getting fixed, you have the blue allele getting fixed. So this is something sort of counterintuitive, right? When the population size is very large, red, which is the advantageous allele, will get fixed, no problems there. But if the population size is small, it's the blue allele that's getting fixed, right? So this is all based on mathematical modeling. And when I read this, I was like, this is so cool. We need to test this empirically, maybe through experiments. But this is a really very, very interesting result. Okay, so they call it reversal of selection, right? It's not, there's definitely drift operating here because the population size is small, but that's not the predominant force. There's something else happening. Stochasticity in population size is doing something which is really quite interesting. Okay, so another interesting work, I'm just going to quickly go over this, again from Gupta lab, is this particular work looks at does mobility really destroy cooperation or aggregation? And basically their work seems to suggest that cooperation plus grouping can co-evolve in mobile populations, and that's because differential cohesion promotes cooperation. So populations show movement, fission-fusion dynamics, you still see cooperation. I'm not going to get into the details of this work in the interest of time. So that's the first part was basically microevolution, but it was all theoretical, model-based result work. However, at CES we've also been doing a lot of empirical work where in the area of population genetics and phylogeography, wherein we sample a species across its range, genetically characterize that species from those samples, and then look at how that genetic variation is distributed across the landscape and what are the drivers of, either population structure or maybe there is no population structure. In fact, my lab, Karthik Shankar's lab, and Sunaga's lab, many such studies have been done. So, but there are quite a few studies, so I'm not going to get into those studies. An example is given here. This is a study on black bugs. We have collected these, the samples were collected from all these grassland locations across India. And if you think about grasslands, these are isolated habitats, right? And they're very few. So what we expected was that there'll be very little gene flow between these grasslands, right? Because these grasslands are separated by human-dominated landscape for thousands of kilometers in some cases. So these grasslands are probably islands, completely isolated from each other. So we expected very little gene flow between them, high population structure. The results were the opposite. This is based on SNP data. I'm sorry, microsatellite data. We do see some structure. There are 3 clusters, one in the north, one in the south, and one in the east. But within each of these clusters, there's a lot of gene flow going on. So somehow these black bugs are able to move between isolated, so-called isolated grasslands, and probably the landscape in between is like a grassland for them because it's all agricultural fields. But anyway, there's a lot of work of this kind also that we have done in CES. But now I'm going to move on to the other interesting aspect of microevolution, that is molecular evolution. This is work from Sunaga's lab. In this study, they looked at the origin of spider toxins, right? Now, it turns out that spiders, the major component of their toxin are something called disulfide-rich peptides, or DRPs, right? And for the longest time, people thought that DRPs, evolved independently multiple times in the spider tree of life. So it was independent evolution of this major spider toxin component in the various spider lineages. At least that was what was thought. But work done in Sunaga's lab clearly established that there was a single origin of this DRP toxin, and then there was multiple diversification events because of gene duplication events, and you have many families or subfamilies of or superfamilies, if I'm right, of DRP proteins or peptides. So they identified 78 novel spider toxin superfamilies, and all of this through phylogenetic analysis, and also provided the evidence for their common origin about 375 million years ago, right? So in spiders, you actually have 2 major groups, the myglomorphs and the arenomorphs. The myglomorphs are, spiders like tarantulas. Their chelicerae go down like that. Arenomorphs are the web-building spiders. Their chelicerae go like that, right? So these are 2 major groups. So basically, their study is suggesting that DRP toxins evolved in the common ancestor of myglomorphs and arenomorphs, right? And another interesting point here was that you have this conserved cysteine framework across toxin superfamilies. So all of these superfamilies of DRP toxins have this conserved motif. So all of which, suggests that it all started from one such peptide or gene which underwent multiple duplication events to give all these families. Okay, here's something else I found really interesting in this work. And Is it a little out of focus or is it okay? Anyway, so turns out the DRP toxins are under positive selection in megalomorphs, right? And they are largely under negative selection in arenomorphs. And one can speculate as to why this is the case, but the authors argue that it's all got to do with prey capture, probably, because the arenomorphs build web, so they use webs to capture their prey, right? Whereas the megalomorphs actually have to inject the venom into the prey and kill it, right? So your venom toxin components are bound to be different. So some of these GRP peptides have been recruited for probably a very different function in case of megalomorphs. So the spider story contrasts with the story of with the venom evolution story with respect to snakes, in the sense that in snakes you have multiple independent origin of many of these toxins, right? And these toxins are not phylogenetically related, whereas in spiders you have this one origin. Now, what can we learn from this study that can be implemented to make sort of better? What happened here? Antivenom. So I'll just briefly talk about that. Many of you already at CATS heard a lot about the work in Sonagar's lab. This is an interesting story where they actually these are elapids, right? And one of the major component of elapid venom are three-finger toxins. And the idea here is to design antibodies against evolutionarily conserved region of the snake toxin, right? Now, the three-finger toxin, this is a sequence from a bunch of different elapids. The dots here represent sequence similarity. So you can see that certain copies of this toxin show a lot of conservation across species. So if you now target those regions to develop your antibody, it's going to be effective against a range of elapids, right? So that's what they have attempted. That's the story behind this particular paper, and I think this work is in full swing now. All right, so I will shift gears and move on to macroevolution, and a lot of this has already been covered by Chanavi. The story is going to be mostly from my lab and Karthik Shankar's lab. And I'm going to concentrate mostly on lizards and frogs because there are so many different groups that we have worked on. Anyway, herbs in peninsular India have both, Gondwanan and African and Asian origin. So many of them are very ancient. Some of them have come into India very recently. All of this we know through all the phylogenetic studies and biogeographic studies that people in my lab and Karthik Shankar's lab have done. The other interesting thing we are observing in peninsular India are endemic radiations. So many of these groups have 20, 30 species confined to peninsular India, and they have emerged from a single Rohit, you're bored. Okay, I think I'll speed it up anyway. Okay, one of the groups are the bush frogs. Amazing radiation of I don't know how many species now. Work done in Shankar lab has eventually resulted in many new species descriptions. And here you see evidence of allopatric speciation, where sister species isolated in adjoining hilltops. You see examples of parapatric speciation, sympatric speciation, amazing group. And within the bush frogs, you have two major clades, and the genetic break that you see in between the two major clades corresponds to Palgard gap. And this is something you see in many other groups where some of these geographic gaps have contributed to genetic structuring of species, of clades, and so on. But there's an interesting caveat to that, which I'll come to in a minute. And bush frogs, I think, are a classic case of adaptive radiation because within this radiation of, I don't know, 50, 60 species, you have different ecomorphs. Some of them are confined to the canopy, others are in the undergrowth, others are on the ground, the others in the stream nearby. So you have all these ecomorphs and they sort of repeat, this pattern repeats itself as you go across Western Ghats. Now, one of the visitors in my lab, Chaitanya, did some very interesting work on Draco. So I'm just shifting gears here just to contrast it with the bush frog story, especially with respect to the geographic gaps, right? Now, Dracos in India, in peninsular India, we believe there's only one species of flying or gliding lizard, Draco dusimiri, right? If you look at the mitochondrial tree, you see multiple clades, and these are all nicely separated. They are all confined to different hill ranges, and here's Palgard Gap that corresponds to this separation here. So one would say, oh yeah, great, Palgard Gap is crucial and has shaped how these haplotypes are distributed along Western Ghats. However, look at the nuclear tree. I mean, the nuclear data. So this is admixture analysis using SNP data. You just see two clusters, two ancestries, right? The northern ancestry starts all the way from Goa, comes till Wayanad, and the southern one is south of Wayanad. Okay, Palghat Gap is here, and there is no separation across the Palghat Gap, right? And there is a admixture zone here. So we suspect that these might be two different species and there's some hybridization going on. But the question is, why this transition from blue ancestry to green ancestry? Right? There is no gap there. There is no geographic gap there. This, I think, is this actually transition zone falls exactly in the region where the climatic regime changes, in the sense that it changes in the south. The dry period length is quite short; it's about 3 months, and from Wayanad onwards, it goes up, right? So there's a change in the climatic rainfall regime as we go across Wayanad. So I think that is what has shaped, at least that's what we believe, has shaped how these, the nuclear diversity is partitioned here. So just looking at mitochondrial DNA can be problematic. Okay, let me go right into the temporal diversification pattern that we see in the various endemic frog groups in Western Ghats. This is again work from Karthik Shankar's lab. These are some of the diversification analysis that Chanvi was also talking about. So basically, they looked at 6 different groups, and there is no support for a model with episodic date shift. So there is, if you have a situation where there's one particular lineage leading to many species, so that would be a sort of an episodic rate shift. There is no evidence of that. Speciation rate slowdown and low extinction in case of Nyctipetracus and Rochesters. Those are the 2 examples there. And constant speciation, very low extinction in the rest, right? So overall, it seems to suggest that even though globally there was a lot of change in climate, starting from Eocene all the way into the Miocene. In the Western Ghats, things were quite stable because we don't see any major rate shifts or change in net diversification rates in these groups. Okay, my last two examples. This is an interesting work where we looked at hemidactylus geckos. We have been working on hemidactylus geckos for decades now. In fact, Rohini, who was my first student, is here. She actually started this work on hemidactylus geckos, and we've been working on them for a while. Today, we have a much better phylogeny of hemidactylus geckos, and this is the global phylogeny of hemidactylus geckos. And we were forever, for a very long time, trying to figure out where this gecko evolved, what was the origin of this gecko. And now with this global analysis, we can say with confidence that it has an Indian origin and dispersal from India into Africa, into Southeast Asia, and from Africa to South America, right? And there are two major sort of parallel radiations, in situ radiations, one in India of about 50 or 60 species, another one in Africa. And both are of pretty much the same age, but the group itself evolved in India. This is a very interesting piece of work on tarantulas, again, a global phylogeny of tarantulas, looking at where this group evolved, why do certain clades have more number of species, and so on. Now, tarantula, there's an interesting distributional pattern. So you have South America with the highest number of species, right? I would say 60 or 70% of all tarantula species are from South America. Then you have Africa and Asia. So if you go by just center of diversity, one would say that South America is the center of origin. But when we did the phylogenetic biogeographic analysis, the results suggested that tarantulas evolved in Africa with multiple dispersal events into India, right? So the center of diversity does not necessarily harbor the origin of that species. Is not necessarily the origin of that species. Now, then the question is, why is there so many species in South America, right? And basically, our what we think is there is something called trait-dependent diversification that's going on in the South American tarantulas. These tarantulas have something called urticating hair. These are anti-predatory sort of defense mechanism, and it's probably this defense mechanism that's helped this group to undergo rapid radiation. So I think that's why you have, by and large, it shows, clade, supports clade age hypothesis. Older clades have more species and so on, but actually, in case of South American species, they have far too many species, and that's probably because of trait-dependent diversification. All right, so I'm just going to wrap it up with and say that the work done in all of our labs here has also contributed to many species, new species descriptions. I think we should count it up. It's probably 100 species or even more. And the data that we have generated, others have used to then describe many new species. So, we made major contribution in terms of characterizing biodiversity and understanding, how this diversity is distributed. Okay, I just wanted to end with this picture. So I told Anubhav that I'm going to surprise him. The microevolution part, I didn't really talk a whole lot about, even though there's a fair amount of work going on, but we are really excited to have Anubhav Khan, who joined us last October. He's our latest hire, and he does population genetics, evolutionary genetics, genomics, and conservation. He's interested in looking at small populations. So microevolution is going to get a big push in CES. Okay, thank you.
Thanks, Praveen.